Hi everybody. So welcome back to the Biochemical Techniques and Tools video lecture series. This is part 2 of the part 2 series. So if you recall from part 1, we looked at how a biochemist may express, isolate, and purify a protein of interest. In our case, we're looking at a protein called Verbosase. And now in part 2, we're going to look at how we can characterize this protein Verbosase using techniques that can help us characterize the structure of the protein, the behavior of the protein, as well as the stability of the protein. If you're really interested in this topic, the class Advanced Biochem, which is taught in the fall, it's a 400 level course, we actually spend the entire semester focusing on just this process. So the biochemical process that goes on in sort of an academic biochemistry lab. All right, so think about taking this course if you found these 2 video lectures very interesting. We are then going to apply the information that we gained from both part 1 and part 2 to activity S6 from our workbook, which is actually a problem-solving challenge. So in this activity, we're kind of given a scenario, we're given some tools at our disposal, and then we're going to further develop some of our problem-solving methodology in order to first define the problem, identify key issues and important issues associated with the problem, figure out how to collect relevant information, determining what is missing, what is needed, identify some assumptions that we need to make, break the problem apart into its pieces, as well as then we'll generate some solutions for these sub-problems, integrate the solutions, test and validate that answer, generalize the solution, and then communicate the solution. So for those of you who've been doing undergraduate research, this whole process should sound very familiar, right? It's sort of what we do in our research projects, okay? So again, just post your muddiest points onto the discussion board if you have any questions, and I will see you in class, and let's get started with part 2.
So now that we've gone over some, very typical chromatography techniques that biochemists use, which techniques can we use to isolate verbosase? So remember, we can use hydrophobicity, we can use ion exchange, we can use affinity, and we can use size exclusion. So in the beginning, remember we talked about verbosase. We know verbosase is a globular protein and it has lots of negative charges on it. So I'm going to draw like a little glob, and he's got a lot of little negative charges on him. So ion exchange could be a good one because we know we have negative charge, so we could use a cationic or a positively charged substrate, stationary phase, but a lot of other proteins have negative charges on them as well. So we may purify some of the unwanted, any of the neutrals or the positively charged proteins, but we're still going to have a lot of other impurities in our target protein. Affinity. Remember I said it binds calcium. Affinity is a great one. As many proteins that are negatively charged, maybe just a fraction of them bind calcium. So I think this is definitely one of the chromatographic techniques that we can use. However, there's still going to be some proteins, some other proteins that are unwanted or untargeted that bind calcium as well. So I think we'll start with a calcium purification, but then we'll follow up with also a size exclusion to be able to get a really pure protein. So the size exclusion we can separate based on size after we've separated by affinity. So first we'll purify out any protein that binds calcium, and then we'll purify the size. Now if we wanted to do more, we could do ion exchange followed by affinity followed by size. Typically you only need to use 2 purification techniques. So why wouldn't you want to start with size? There's going to be a lot of proteins that are in that 19 kilodalton range, and so the size exclusion is not as effective until you do some other separation technique before it. There's probably not very many other proteins at 19 kilodaltons that bind calcium. So we did our calcium affinity chromatography, and now we've ran our size exclusion, and here is our spectra. So our large proteins are going to be our first few peaks, and then our smaller. So we have large proteins here, small proteins here. Now, earlier in the lab, I ran a ladder of proteins with various sizes, and I found that proteins around 18 to 20 kilodaltons are going to come off in the range of 7 to 9 minutes. And lo and behold, we have a very nice peak here at about 8 minutes, which is peak 3. So I'm going to predict that this is around 19 kilodaltons, that this is verbosase. Now again, as a good biochemist, I'm still going to collect and save all of these fractions, because if this isn't verbosase, one of these are. So how are we going to determine, is peak 3 verbosase? So there's two ways we can do this. We can determine it using gel electrophoresis or using mass spectrophotometry. And we can utilize this technique not just to determine identity, but also to determine our purity of our protein as well. So let's talk about page analysis. So gel electrophoresis is sort of like chromatography in that we're separating molecules based on some sort of physical property. And instead of using silica gel, we're using, for proteins, often polyacrylamide. Now, depending on how much of this polymer of polyacrylamide we have, we can separate different size ranges. Higher percentages give you better resolution for small proteins, while lower percentages give you higher resolution for large proteins. And when I say resolution, I mean amount of separation. So good separation, good resolution. So as you see here, we can have two forms of page analysis. You can have native page analysis, where the protein is ran without the presence of denaturants, and that keeps the protein folded into its tertiary structure. Denatured means that the protein is ran with denaturing agents to unfold the protein. Here we can see we're doing a polyacrylamide gel electrophoresis of a native protein. You have a negative charge at the top of the gel and a positive charge at the bottom, so molecules will separate based on their charge and their size in native gel. Now we can overcome the charge of the molecule by adding a molecule called SDS. And what SDS does is it comes in and it surrounds the protein, and it can unfold the protein to give the protein an overall negative charge. And since the ratio of the size to the charge will be the same, now all the proteins in the gel will separate merely on size instead of having to deal with the size and the charge ratio. So, what you do, again, remember we have our negatively charged electrode and our positively charged electrode, and then here is your polyacrylamide gel. Alright, so this has got your polyacrylamide polymer in there that the proteins have to wiggle around and get around and go through. So the larger the protein is, the slower it migrates through the gel versus smaller fragments. So you have wells that you add your protein to. You turn the electric current on, and so the proteins are pulled, since they have a formal negatively charged, down to the bottom of the well, and then based on size, separate. So we ran some SDS page of the verbosase column fractions, and we can look at that here. So M is a marker. So this is a standard protein ladder of proteins at very specific lengths. So now we have something to compare our unknown proteins to identify their lengths. So this first lane, as they're called, is the total cell lysate. So these are all the proteins that was in that aqueous layer after we lysed the cells and spun it down. So you can see there's a ton of different proteins here. Then we loaded that lysate onto that calcium stationary phase, and anything that didn't bind to that stationary phase is here in this lane. So we can see a lot of proteins that didn't bind the calcium just flowed through. Then we eluted all the proteins that did bind to the calcium. So we flooded the system with calcium, which pulled the proteins from the stationary phase into the mobile phase. And we can see that here. So if we compare these two fractions, we can see that we got much better purity. So now we don't have nearly as many proteins as we did in the original lysate. And look, we have a nice big band right here between 25 and 15. Could that be verbosase? That is around 19 kilodaltons. I don't know. So then we did our size exclusion, and remember we like that peak 3, that was around 8 minutes. So from running some standards that are around 19 kilodaltons, we found that they came out around 8 minutes. And indeed, look at this beautiful clear band. So that 8-minute peak, I collected 2 different fractions of, and so I wanted to run both fractions. And both, as you can see, are very pure. We do not see any other protein bands. And based on the intensity of this band, I can say that we have a very good concentration or lot of pure, beautiful verbosase. This is awesome. Great science, guys. Now, we want to further identify the molecule. Okay, so gel electrophoresis told us, all right, we do have a band. It's at about 19 kilodaltons. And it looks pretty good. I mean, chances are it is verbosase, but, you know, we're scientists. We want to verify. So to do that, we're going to run mass spec. So mass spec instrumentation differs by ionization method and the separation method. Commonly, for proteins, MALDI-TOF, and follow this link and you can learn more about MALDI-TOF, is utilized. So what mass spec does is it's going to separate molecules based on their size, but also based on their size to charge ratio. As we see here, TOF analysis, which stands for time of flight, separates molecules based on the time that it takes them to fly through the cylinder and hit the detector. So once the sample is inserted, the sample gets ionized, and it's sent through, or it's flying. They like to say that you make the molecule, or you make the ion fly, through a long cylinder. Now, lighter ions are going to travel faster and hit that detector sooner, while heavier ions travel slower and hit the detector later. So determining that time will identify the molecular weights. From our results, we see that we get a very pure sample because there's only one peak, and that peak just happens to be at 19,198 daltons, or 19.2 kilodaltons. Beautiful. We can further use mass spec to fragment this protein amino acid by amino acid and sequence it to then verify that this is verbosase. Which, we'll skip ahead. Yes, we did it. It is verbosase. Yay! We have very pure, lots of verbosase enzyme now to analyze. So now what? So now we can begin our characterization. So we've probably spent a month just expressing the protein, growing it up in the E. coli cells, and then purifying it. And so now, after a month of all that hard work, we can actually begin our experiments. So there's lots of things you can do with proteins. We can obtain a structure. What is the tertiary and secondary structure of verbosase? Because we know structure is function. Well, there's 2 ways you can do that. You can figure out the structure in solution, or in a buffer using NMR. Again, follow this link if you want to find out more about how NMR, or nuclear magnetic resonance spectroscopy, is used to determine a structure. Y'all should remember this from organic chemistry. I know y'all did a little bit of NMR there. You could also make a crystal of the protein, which is relatively difficult to do for proteins, but it is doable. It's been done many times before. And you can determine the structure using X-ray crystallography from the diffraction patterns of X-rays hitting the atoms. We could also characterize the binding of calcium. So remember, we know verbosase binds calcium, but we don't know how it binds calcium. We don't know the characterization of it. So we can do that using isothermal titration calorimetry. This can tell us the change in enthalpy, the change in entropy, as well as the change in Gibbs free energy upon binding of calcium to verbosase, as well as an idea of the binding affinity and the number of binding sites. Does verbosase bind 1 calcium, 2 calciums, 10 calciums? We don't know. That's something new for us to discover. Isothermal titration calorimetry is a lot like bomb calorimetry, or if y'all ever did the coffee cup calorimetry experiment where you put antacids into acetic acid and then measured the temperature changed upon the reaction, it's sort of like that. So when calcium binds to verbosase, there may be heat released. And so this instrument can measure the amount of heat released or absorbed upon that interaction. Again, this can tell us if the reaction is endothermic or exothermic, and that can tell us a lot about how this protein behaves in the body. We can also characterize the thermodynamics of unfolding the protein, and that can give us an idea of the protein's stability. So we can do this using differential scanning calorimetry, which will tell us the melting temperature, the change in enthalpy, entropy, and Gibbs free energy of unfolding. And then we can figure out, well, does this change in the presence of calcium? Does the binding of calcium stabilize the protein structure or destabilize the protein structure? Is it easier to melt or is it harder to melt? And so differential scanning calorimetry, what that does is you put the protein in and you slowly heat this system up. And the system constantly monitors the change in heat or the change in the heat capacity of the protein as we heat up. And if there is a change, it will start to plot that change. And again, this is more detail that we can get into, but is it an endothermic or an exothermic event when the protein unfolds? 9 times out of 10, when proteins unfold, it is an endothermic event. It requires a lot of heat to do this, because the protein would rather be folded than unfolded, again, shielding those hydrophobic residues while increasing favorable intermolecular interactions. Also, does it aggregate in the presence or absence of ligand? So we can determine, if we add calcium, does it cause the protein to oligomerize or aggregate? Do we get subunits coming together? Or is it already aggregated? I don't know. So analytical ultracentrifugation can tell us, based on the diffusion through a gradient in a centrifuge, if verbosase aggregates. So we can see if it aggregates, say, sort of like hemoglobin, right, where you have 4 subunits. If it aggregates like hemoglobin, we would see it diffuse differently as a quartet than it would as a monomer, because those are going to have different density profiles. It's pretty interesting. So what have we learned about verbosase? Oh, we learned so much. So first off, from NMR, we were able to get the structure of one verbosase molecule. And it looked like this guy. So we had some alpha helices, we had some beta rolls, all sorts of interesting things. However, from analytical ultracentrifugation, we found that this molecule did not exist as a monomer in the presence of calcium, but it existed as a 1, 2, 3, 4, 5, 6, 7 heptamer in the presence of calcium. Without calcium, we see that it exists as only a monomer. So we see that this oligomerization falls apart in the absence of calcium. We also discovered that this molecule will bind 1 calcium per monomer. So in the heptamer, we have 7 molecules of calcium bound, 1 for each unit. We learned that the binding affinity is very high, so it loves calcium, it binds it very strongly, and that it is an exothermic interaction, so it's favorable. The binding of calcium to the protein is very favorable in exothermic, releasing heat. And we also saw that upon binding of calcium, the Tm increased and the delta G decreased. So we know that the stability of the molecule increases when calcium is bound and so does the propensity for oligomerization. So now that we've learned all of this new stuff about verbosase, well, what can we do with this information? So now that we've done all this, now we want to say, all right, how can we reduce the amount of verbosase in all of these politicians and overly talkative professors so that students can finally get out of the classroom on time and not be a half hour over anymore and go do their homework? So we decided, well, let's look at, can we disrupt the oligomerizing interaction? So we saw from our solution structure that the oligomers interact at these regions here. So if we mutate these regions to give them each negative charges or positive charges so that they repulse each other instead of attract each other, would that disrupt the oligomerization and then decrease the activity of this enzyme? Could be. So what do? Well, we start all over from the beginning. We change the gene sequence so that those amino acids are mutated. We insert the mutant gene. We insert that plasmid. We regrow the protein. We relysate and spin it down. We repurify for the new protein. And we do all of our analyses over again and then recharacterize the mutant. It's a lot of fun, and there's always a lot of things to do in a biochemistry lab. So if y'all have any questions, I will see you in class next time. Feel free to email, message me on Facebook, and I will see y'all then.